Okay, let's unpack this. Imagine a brilliant scientist, Paul Ehrlich, right? A pioneer in immunology. And he's so disturbed by an idea that he actually called it horror autotoxicus.
Yeah, the horrifying thought of your own body turning against itself.
Exactly. And for decades, this whole concept was argued about, even kind of suppressed in scientific circles. But today, well, we know it's not just possible, it's a profound reality. It affects millions of people. So we're going to dive deep into this central mystery. How does our immune system, which is designed to protect us, right, from invaders, how does it sometimes just go rogue and mistakenly attack its own host?
Well, what's truly fascinating here, I think, is how close Gerlich's later interpretation was to how we see it now. He came to believe the immune system should exert control over itself.
Ah, okay, so control.
Exactly. And that precise control, or, the critical lack of it, that's really what we're exploring today.
Right.
Our deep dive today, it's kind of built around three pillars, three interconnected ideas: immune tolerance, autoimmunity, and transplantation.
And why group those three together?
Well, they really are a natural grouping. Because genuinely mastering one area, doesn't matter if you're a clinician or a researcher, it requires a fundamental grasp of how all three interact. You see, immune tolerance, that's your body's really sophisticated ability to tell self from non-self, and even from beneficial companions like gut microbes.
Okay.
Now, when this delicate tolerance system breaks down, that's when we get into autoimmunity, the immune system attacking its own tissues. And then in transplantation, the immune system's inherent drive for self-tolerance, that actually becomes the main barrier to accepting life-saving foreign organs.
So understanding one helps unlock the others.
Precisely.
Okay. So our mission for you, listening in, is to provide a shortcut to help you get truly well-informed on these topics, almost like, an elite professional in the field.
Yeah, so you can really master these complex concepts, connect the crucial dots.
And gain a profound appreciation for how incredibly balanced your own immune system really is, most of the time anyway. We'll be pulling apart the intricate mechanisms, looking at the history, dissecting the different diseases, and exploring those cutting-edge treatments that are, well, shaping the future of medicine. So to really get why our body sometimes turns against itself, we first have to grasp this incredible daily tightrope walk of immune tolerance. Let's start right there with that cornerstone concept.
Indeed. So when we talk about tolerance in the immunology, what we mean is the immune system's multi-layered protection against reacting to its own host components, your own cells, your tissues, and importantly, even against benign companions like the food we eat or, those vast populations of beneficial gut bacteria.
Right.
Historically, our understanding of tolerance was, well, pretty simplistic. For a long time, the main idea was that tolerance happened mostly through elimination, getting rid of self-reactive cells. Kind of an ignorance is bliss model.
So the thinking was just delete anything dangerous.
Pretty much. If the immune system didn't see self-antigens, or if any cells that could react to self were quickly weeded out, then autoimmunity wouldn't be a problem, like a quality control system just discarding anything potentially faulty.
So if it looked even slightly off, just gone.
Exactly. But contemporary studies, especially over the last maybe 2 decades, they've given us really compelling evidence for a much more nuanced, much more active role.
Ah, so not just passive deletion.
No, not just ignoring or passively eliminating. It's about the immune system actively recognizing and protecting self-compounds and beneficial microbes. And this shift in understanding, from passive ignorance to active engagement has been revolutionary.
How so?
Well, it's led to the characterization of regulatory T cells, or TREGs. These are a special type of lymphocyte, a white blood cell, that, kind of surprisingly, recognize self-proteins with high affinity. But then they inhibit the immune response rather than kicking one off.
Wow, so they see self but calm things down instead of attacking.
Precisely. And that critical discovery alone, it's opened up entirely new lines of investigation and potential therapies in tolerance, autoimmunity, and transplantation science.
And it sounds like this isn't just a shift in immunology thinking, but maybe it mirrors a broader change in science, moving from simple models to understanding these complex, dynamic, interconnected systems.
That's a really excellent observation, yes. This shift definitely reflects a deeper appreciation for the intricate choreography within biological systems. It's not just about one thing happening or not, but about these complex balances and active negotiations within the whole system.
Okay.
So before we really dive into the core mechanisms of this active tolerance, let's touch on a fascinating first line of defense. How the body sometimes just prevents the immune system from even seeing some self-antigens, basically hiding them. This is called antigen sequestration.
Hiding its own antigens. How does it manage that? It sounds like a stealth tactic.
It is, in a way. Sequestration, or partitioning, it involves physically isolating certain antigens from immune cells. A classic example is the anterior chamber and lens of the eye.
Ah, okay. The eye.
Right. These are considered privileged sites because they have very little or maybe even no lymphatic drainage, so the tissue-specific antigens expressed there. They're largely isolated from interacting with most parts of the immune system. Under normal circumstances, this lets these antigens evade encounter with reactive lymphocytes, minimizing the chance of an immune response.
So if the immune system doesn't see it, it can't react. Makes sense. But what's the downside? What happens if that barrier gets breached?
That's the crucial consequence.
Yeah.
If these physical barriers are breached, let's say, by trauma or injury to the eye, these newly exposed antigens can suddenly be seen as foreign.
Because the immune system never learned they were self?
Exactly. Having never learned to tolerate them, the immune system can then attack aggressively.
Yeah.
This leads to locally damaging inflammation, tissue destruction, maybe impaired vision, and what's even more striking, sometimes trauma to one eye can cause inflammation in the other eye.
Seriously?
It's likely due to clones of recently activated immune cells now exposed to these previously hidden antigens circulating systemically, right? And then attacking the other eye. It's called sympathetic ophthalmia. It's a powerful demonstration of how important that initial context of exposure really is.
But if these sites are truly privileged, how do we explain, say, recent findings about the central nervous system? I've heard it's not as completely cut off as we once thought.
You're hitting on a critical nuance there.
Yeah.
Our initial view of immune-privileged locales might have been a bit oversimplified. For instance, recent research. It's shown that the CNS, the central nervous system, it does get regular visits from some circulating lymphocytes, and it does have lymphatic drainage, although it's specialized. Plus, there are antigen-presenting cells living right there in the CNS. And we know many self-reactive lymphocytes, even ones that recognize nervous system bits, can be found in healthy people.
So does that mean active suppression is maybe more common than we thought, even in these privileged places?
It strongly suggests that yes, that these protected sites might not rely on absolute barriers, but maybe they're structured in ways that, under normal conditions, bias immune engagement towards tolerance rather than assault. A more sophisticated kind of control. Maybe they have partial barriers like the blood-brain barrier. They can open and close as needed.
So it's not just about hiding things away, but also actively training the immune system. How does the immune system train its cells not to attack self, right from day one, like an immune cell boot camp?
Absolutely, that's a great analogy. This foundational training happens through what we call central tolerance, and it occurs mainly in the primary lymphoid organs, the thymus for T cells and the bone marrows for B cells.
Okay, thymus and bone marrow.
And this process, it involves a kind of dual approach. First, encouraging the destruction or elimination of potentially harmful self-reactive lymphocytes. And second, at the same time, cultivating or engaging certain other self-reactive cells specifically for the protection of self-structures.
Right, let's start with the elimination part. Our immune cell receptors, they're generated through random genetic shuffling, right? So it seems almost inevitable some would end up recognizing self.
That's precisely right. The genetic rearrangement of T cell receptors, TCRs, and B cell receptors, BCRs, it involves random nucleotide additions. This means variable regions capable of reacting with self-antigens are just an unavoidable consequence of generating all that immune diversity.
So if all those self-reactive cells matured,
autoimmune disease would be rampant. Instead, most developing lymphocytes with receptors that recognize self-antigens with high affinity, they get eliminated in the thymus and bone marrow before they mature. This crucial process is called negative selection.
negative selection, got it.
It induces apoptosis that's programmed cell death in those developing lymphocytes that bind too strongly to self-antigens expressed right there in those primary lymphoid organs. It's a pretty ruthless culling of the potentially dangerous ones.
But how do the cells in the thymus, for instance, know about self-antigens that are specific to, say, the pancreas or the liver? That seems like a huge information challenge for just one organ.
That's where this remarkable transcription factor called AIR comes in. AIR stands for autoimmune regulator.
AIR, okay.
What AIR does is it allows certain epithelial cells in the thymus, in the medulla, to express tissue-specific self-antigens from pretty much every other organ in the body.
Wow, that's incredible. So the thymus gets a preview of the whole body.
Kind of, yeah. It's an amazing mechanism.
Ah.
It allows for the deletion of potentially harmful self-reactive T cells that might otherwise attack those specific tissues, even though those tissues aren't physically present in the thymus.
And if air goes wrong,
Well, mutations in the R gene are directly linked to severe systemic autoimmune syndromes, things like APS1. It clearly demonstrates just how critical ARI is for central tolerance.
that's a really stark example. Are there other classic experiments that show this elimination process or how the system deals with self-reactive cells?
Oh, yes. Christopher Goodnow and his colleagues did a really classic experiment using transgenic mice. These mice were engineered to express a protein called hen-egg lysozyme, or HEL, basically everywhere as self. And crucially, they also had a transgenic B cell receptor specifically for HEL.
So B cells designed to attack HEL in a mouse where HEL is self. What happened?
What they found was that these self-reactive B cells were either removed or inactivated after they encountered the HEL self-antigen. The mice were basically rendered incapable of attacking HEL. It was direct evidence for elimination or inactivation.
Okay.
And then David Nemesi's group showed that some developing B cells actually have this clever salvage pathway called receptor editing.
Receptor editing. They can change their receptor.
Yeah, essentially.
Mhm.
During central tolerance, if a B cell makes a self-reactive receptor, it can sometimes re-engineer it. It swaps out a problematic V-region gene segment through more genetic recombination. This often produces a less autoreactive receptor, letting the cell avoid elimination. It effectively edits out its self-reactivity, giving it a second chance.
So it's not just deletion. Some get a do-over to become non-self-reactive.
Yeah.
But you also mention a more proactive side, engagement or cultivation of self-reactive cells for protection. That sounds counterintuitive. Why would the immune system choose self-reactive cells?
It does sound counterintuitive, but it's a critical part of this newer understanding of central tolerance. Some lymphocytes with high affinity for self-antigens are actually selected for survival during development instead of being eliminated.
Okay.
Specifically for T cells in the thymus, these selected self-reactive cells start expressing the FOXP3 transcription factor
FOXP3, that's the marker for.
regulatory T cells, or TREGs. Once these cells leave the thymus, they're known as thymic-derived regulatory T cells, or Treg cells. And their crucial job is to actively suppress or regulate autoimmune responses out in the periphery, in the rest of the body.
But if they have high affinity for self, just like the ones that get deleted, what determines their different fate? What's the subtle signal that says become a regulator instead of self-destruct?
Yeah, that's the nuanced part, and honestly, it's still an active area of research. Based mostly on mouse studies, it seems to be a mix of both positive and negative signals during their development. Things like intercellular interactions, CD28 binding CD8-086, for example, or the presence of certain cytokines in the local environment can sway the outcome. There's this hit-and-run model proposed for T-cell receptor engagement in the thymic medulla. The idea is that short, high-affinity, but transient binding of the TCR to the MHC antigen complex. That seems to favor generating these regulatory cells.
Okay, a quick hit.
Right. Whereas a more sustained, high-affinity TCR engagement seems to favor deletion. So it's about the precise quality and maybe the duration of the signal. It's a very delicate dance of molecular interactions.
So those Treg cells leave the thymus, go out to do their job. Are there CD8 plus versions of these regulatory cells too?
Yes, there are. Though they're a smaller population and, frankly, less well-characterized compared to the more common CD4 plus Treg cells. Conventional CD8 plus T cells recognize antigens using classical MHC molecules, right?
Right.
But the best characterized CD8 plus Treg cells, they often seem to be restricted to non-classical MHC class I molecules, like Q1 in mice or HLA-E in humans. These molecules are involved in presenting things like lipid antigens.
And are they important?
Seems so. Studies in mice lacking these QA1 restricted CD8 plus T cells showed they developed aggressive autoimmune reactions. This suggests they're involved in regulating CD4 plus T cell responses and maintaining peripheral tolerance. They've also been shown to induce tolerance to heart transplants in rats and protect mice from EAE, that animal model of multiple sclerosis. So yes, they seem crucial even if we understand less about them.
Okay, so even with this really elaborate system of central tolerance, it makes sense that some self-destructive lymphocytes might still slip through the net. What happens to those that escape the boot camp and get out into circulation?
That's exactly where peripheral tolerance comes in. It provides multiple additional safeguards outside the primary lymphoid organs, outside the thymus and bone marrow.
Okay, backup systems.
Exactly. These mechanisms mainly operate in secondary lymphoid organs, think lymph nodes, spleen, or sometimes directly at the tissue site where the relevant self-antigen is expressed.
And why do cells escape central tolerance in the first place?
Well, a couple of reasons. One, not all self-antigens are expressed in the central lymphoid organs, even with air doing its job. And two, there's an affinity threshold for clonal deletion, meaning some clones that are only weakly self-reactive might survive that initial weeding out process.
So these peripheral mechanisms then have to limit or redirect the activity of these potentially harmful escapees. How does the immune system decide to induce tolerance out there instead of launching a full attack? What's the deciding factor?
The context. The context in which the antigen is presented is absolutely critical. Antigens that induce tolerance are called tolerogens, not immunogens. And the fascinating thing is, the same chemical compound can be both an immunogen and a tolerogen. It depends entirely on the context of its presentation.
Okay, context is key.
Right. So for a T cell that gets engaged by a tolerogen or just in a tolerogenic setting, there are typically two fates other than just dying off energy, which basically means unresponsiveness.
Like it gets switched off.
Switched off, exactly. Or regulation, where the engagement actually leads to suppression. Naive T cells that enter this regulatory pathway out in the periphery, they can be induced to express FOXP3 and become Treg cells, peripherally induced regulatory T cells. They also act as antigen-specific inhibitors of activation.
So it's not just what the antigen is, but the environment or the way the immune system first sees it that can fundamentally decide between tolerance or attack. That's a really powerful idea. What are some of the factors in that context that push towards tolerance?
Exactly. Well, besides things like exposure during fetal or early neonatal life, when the immune system is still setting its tolerance rules, other factors that promote tolerance include things like high doses of antigen or long-term persistence of the antigen in the host. And critically, the absence of adjuvants. Those are compounds that normally enhance immune responses, often signaling danger.
Right, like in vaccines.
Precisely. Also, low levels of co-stimulation or antigen presentation by immature or unactivated antigen-presenting cells, APCs, those tend to promote tolerance. Think of it like this. For a full-blown pro-inflammatory response to happen, you need this precise, coordinated choreography between multiple cell types, right? An activated APC has to present the antigen to a naive CD4 plus T cell, which then coordinates with B cells and CD8 plus T cells.
Right, whole team effort.
Yeah. So if one or more of those crucial dancers are missing, or maybe present but in an inactivated state, tolerance is often the outcome. It's like a complex performance, and missing a key performer can stop the whole show.
So who are the specific players on this tolerance team out in the periphery? What cells are mainly responsible for suppressing these self-responses?
Well, it's likely that many circulating B and T cells that have some cell specificity actually have a regulatory function. It's truly a balance, a teamwork situation, and we're probably still identifying all the members of this team. But the most well-characterized are definitely the regulatory T cells, the Tregs, which includes subsets of both CD4 plus and CD8 plus lymphocytes.
Okay, Tregs again.
Right. Remember, for a T cell to get fully activated, it needs two signals. Signal 1 is the T cell receptor binding the antigen MHC complex. Signal 2 is co-stimulatory engagement, like CD28 on the 2 cell binding to CD80 or CD86 on the APC.
Right, the two-signal hypothesis.
Exactly. And Mark Jenkins and his colleagues showed in some early, really important experiments that if you stimulate CD4 plus T cell clones in vitro just through the TCR signal 1 only, without that second co-stimulatory signal, they become anergic, unresponsive.
And this led to finding CTLA-4, right? That molecule that acts like an off switch.
That's right. Looking closely at co-stimulation led to discovering CTLA-4. It's related to CD28, but when it binds CD80-86, it actually inhibits T cell activation. We now know there are many such molecules delivering supplementary signals, often called immunomodulatory.
And CTLA-4 is really important.
Oh, hugely. The evidence is stark. Mice engineered to lack the CTLA-4 molecule. They display massive lymphocyte proliferation and widespread lethal autoimmune disease. It clearly shows its essential role in maintaining peripheral tolerance.
Wow. Okay, so what are the key characteristics, the hallmarks of these CD4 plus Tregs, and how do they actually do the suppressing?
To identify them, CD4 plus Tregs are typically characterized by expressing that FOXP3 transcription factor we mentioned, plus high levels of CTLA-4 and also high levels of the IL-2 receptor alpha chain, which is called CD25.
Okay, FOXP3, CTLA-4, high CD25.
Right. And remember, these can originate in the thymus as T-Tregs, or be generated out in the periphery as Tregs from naive non-regulatory precursors. The critical importance of FOXP3 expression, which seems both necessary and sufficient for their immunosuppressive function, is seen tragically in humans with mutations in this X-linked gene. It causes a severe multi-organ autoimmune disease called IPEX syndrome.
Okay, so they're clearly vital. How do they inhibit immune responses? What's in their toolkit?
They've got a sophisticated, multi-pronged arsenal. They use both contact-dependent and contact-independent mechanisms. They can actually directly kill APCs or effector T cells using molecules like granzyme and perforin.
Wow, direct killing.
Yeah. They also modulate other cells through surface receptor engagement. A prime example is CTLA4 on the TREG interacting with CD8086 on the APC. This can lead to several things. The APC might reduce its expression of those co-stimulatory molecules, making it less activating. It can also decrease the APC's production of pro-inflammatory cytokines like IL-6 and TNF-alpha. And it can activate an enzyme in the APC called indoleamine 2,3 dioxygenase, or IDO.
IDO? What does that do?
IDO converts tryptophan into kynurenine, which creates a very immunoinhibitory local environment. So basically, the TREG can disarm the APC or even turn it into an immunosuppressive force itself.
That's clever. What about other methods?
Well, TREGs also secrete powerful immunosuppressive cytokines, primarily IL-10, which is a major one, but also TGF-beta and IL-35. These suppress the activity of nearby T cells and APCs. And here's another neat trick. Because TREGs express high levels of CD25, that high-affinity IL-2 receptor, they can act as an IL-2 sponge.
My IL-2 sponge.
Yeah. They soak up this crucial cytokine, IL-2, which is needed for the growth and survival of effector T cells. By grabbing all the available IL-2, they effectively discourage the expansion of local immunostimulatory effector T cells by starving them of this vital resource.
That's quite an arsenal. Is there strong evidence that these CD4 plus TREGs really control autoimmune responses in, actual living organisms?
Oh, absolutely. There are great examples. Experiments in non-obese diabetic, or NOD, mice and biobreeding, or BB rats. These are two strains that are prone to developing autoimmune diabetes spontaneously.
Right.
Injecting these animals with normal CD4 plus T cells, especially the CD25 plus subset, which is enriched for TREGs, significantly delayed the onset of diabetes.
Okay, that's compelling.
And similarly, in mice that genetically lack CD4 plus TREGs, inflammatory bowel disease is very common. And if you deplete this TREG subset in normal mice, it makes them susceptible to experimental autoimmune encephalomyelitis, or EAE, that model for NHIS, which they would normally be resistant to.
So they're key for gut health and preventing EAE mess-like disease, too.
Exactly. It highlights their importance in maintaining systemic homeostasis, particularly in relation to our commensal microbes in the gut. The key takeaway here is that TREGs don't just use one trick. They employ this sophisticated, multi-pronged strategy to actively suppress inappropriate immune responses. They really are the immune system's internal peacekeepers.
And that leads us to a fascinating concept I've heard mentioned. Linked suppression. How does that work? Where TREGs seem to suppress responses to other unrelated antigens nearby?
Linked suppression.
Yeah.
That's a really interesting phenomenon, and it shows the broad reach of TREG influence. While TREG inhibition is generally antigen-specific, they recognize a particular self-antigen experiments have shown that FOXP3 plus T cells can inhibit other T cells nearby that are recognizing different antigens.
How does that happen?
It seems to occur when a single APC simultaneously engages both a TREG, recognizing its specific antigen, and a bystander T cell, recognizing a different antigen.
Okay, one APC talking to both.
Right. The TREG then transmits inhibitory signals to that APC, using both contact-dependent methods like CTLA4 and contact-independent factors like IL-10 or TGF-beta. And this inhibition can then spread to include that bystander T cell, partly through those soluble inhibitory factors, and partly because the APC itself gets decommissioned, reducing its ability to stimulate any T cell.
So the TREG dampens the whole local environment via the APC.
Exactly. This simultaneous processing of different antigens might happen naturally in vivo, say, if the antigens are part of the same pathogen, but it doesn't strictly require that. Linked suppression provides another crucial way. TREGs support local self-tolerance in tissues, even if there aren't strong danger signals around, preventing a wider immune flare-up.
Now, it's not just T cells involved in this whole regulatory network, is it? I've heard B cells can also act as regulators.
You're absolutely correct. Regulatory B cells, often called Bregs or sometimes B10 cells, do exist, and they play an important role in maintaining tolerance as well.
Bregs or B10 cells. Why B10?
Mostly because while they lack really clear consensus phenotypic markers that uniquely identify them, A key feature of most identified Breg populations is that they produce high levels of the inhibitory cytokine IL-10. So B10 often refers to IL-10 producing B cells.
And what do they do?
They seem to suppress inflammatory cascades, for example, those associated with another cytokine called IL-1. There have been mouse models of autoimmune diseases like MS and RA where if the animals were engineered so their B cells couldn't secrete IL-10, they actually developed chronic Th1 cell activation and a worsening of their disease.
So Bregs are important inhibitors too.
Yes, and while B cells aren't the only cells making IL-10, it's clear Bregs are important players in inhibiting adaptive immunity. It also shows something else interesting, how the maintenance of T cell tolerance to self-antigens actually helps enforce B cell tolerance to those same antigens. They're all interconnected.
Are there any other accessory cells helping out with regulation?
Yes, definitely. For instance, TREGs can, as we touched on, decommission APCs. They signal APCs to reduce their co-stimulatory potential, like decreasing CD80-86 expression. This not only stops that APC from stimulating effector T cells, but might even encourage more TREG production when naive T cells interact with that now tolerogenic APC.
Okay. Any other cell types?
Another important regulatory immune cell type is the myeloid-derived suppressor cell, or MDSC. These are actually a pretty heterogeneous group of immature myeloid cells. They were first identified in pro-tumor microenvironments, where they help tumors evade the immune system.
Right, in cancer.
But more recently, MDSCs have gained recognition in situations of chronic inflammation, including some autoimmune diseases. They seem to suppress local antigen-specific T cell responses, partly by secreting powerful inhibitory compounds like IL-10, IDO, arginase, nitric oxide, and also by putting up suppressive flags or ligands on their surface like PD-L1, essentially telling T cells to calm down.
You mentioned gut commensals earlier, and this seems like a good time to circle back to that really hot topic. It takes guts to be tolerant.
Yes, this is a truly significant area of appreciation that's really blossomed in the last decade or so. Our microflora, especially in the gastrointestinal tract, play an absolutely crucial role in regulating the immune system.
How so?
Well, the commensal organisms living in our gut, our microbiota, they actively induce tolerance to themselves, which makes sense. But perhaps even more importantly, they seem to be key players in maintaining a level of systemic immunological balance, a balance that allows for the development of tolerance to self, while also enabling effective identification and elimination of actual pathogens.
So it's a two-way street.
Absolutely. It's a two-way communication. The microbes get a place that thrives, and the host benefits from immune health. For instance, germ-free mice raised with no bacteria at all, they show defects in both humoral and adaptive immunity. And in some strains, they have an increased susceptibility to autoimmunity. And a recent study even found a paucity, a lack, of certain specific bacterial species in the intestinal flora of people in the very early stages of human rheumatoid arthritis. These kinds of observations strongly suggest a clear influence of our commensal microbes on the induction, or maybe the severity, of some autoimmune diseases. This connection between our gut and our overall immune health is truly fascinating.
It really is profound. So the gut bacteria aren't just passive bystanders, they're active participants in keeping our immune system balanced. How do they actually do that?
Well, one mechanism involves specific molecules produced by the bacteria, like polysaccharide A, which comes from a common gut bacterium called Bacteroides fragilis. This molecule alone was shown to restore immune balance in those germ-free mice.
Just one molecule from one bug?
Yeah, it clearly demonstrates that a single microorganism, or even just a single molecule from a microbe, can have these systemic effects on immunity. And this leads to the microflora, or altered microbiota hypothesis. It basically posits that changes in our gut flora, maybe due to diet or antibiotic use, might disrupt these normal microbially mediated pathways that are important for immune tolerance. There's a clear link being drawn now between the state of the intestinal microbiota and gut health, including conditions like inflammatory bowel syndromes.
And how does the immune system in the gut itself play into this?
Well, intestinal epithelial cells, the cells lining the gut, and also mucosal dendritic cells, DCs, that live just below them. They express several types of innate receptors, like toll-like receptors, TLRs, and NOD-like receptors, NLRs. These receptors sense the microbial environment.
Okay.
Mucosal DCs are known to actually sample the contents of the intestinal lumen, reaching through the epithelial layer sometimes. They can be a crucial connection between the commensal microbes and the maintenance of tolerance. For example, mice lacking the MyD88 gene, which encodes a key intracellular signaling molecule used by many of these receptors were found to be more susceptible to intestinal injury and autoimmunity.
So, signaling through these innate receptors, which we usually think of as triggering inflammation, sometimes it induces tolerance instead.
Exactly. It suggests that in some contexts, especially with signals from our normal gut microbiota, signaling through these pathways can deliver tolerogenic signals, possibly instructing DCs to promote TREG development. It's a really active area of research. And clinically, things like fecal transplants are even being used trying to rebalance immune activity by restoring a healthy gut microbiome.
That's incredible. And speaking of surprising connections, what about the idea that we are what we eat extends right down to our immune system?
It absolutely seems to. Here's a really compelling example. In vitro studies, so studies in lab dishes, have shown that just adding sodium chloride common table salt to cultures of CD4 plus T cells can actually drive the development of Th17 cells.
Salt drives Th17 development.
In vitro, yes. And remember, the expansion of Th17 cells has been linked to the development of certain autoimmune diseases. So even something as fundamental as our diet, like salt intake, could potentially directly influence pathways involved in immune tolerance and autoimmunity.
Wow. Okay. So putting this all together, what does it mean? It sounds like tolerance is this constant, active, daily process, almost an uphill battle for our immune system.
That's a good way to put it. It's a dynamic balance.
And when that battle is lost, when that balance tips, that's when we enter the realm of autoimmunity.
Precisely. Autoimmune disease, simply stated, is caused by the failure of these intricate tolerance processes we've just spent time exploring. It leads to the immune system attacking the body's own self-tissues.
Yeah, it's common, right?
Unfortunately, yes. According to the NIH, up to maybe 8% of the population is affected by one of more than 80 different autoimmune diseases. And these conditions, they're often chronic, often debilitating, leading to significant morbidity, reduced quality of life, and sometimes even mortality from complications or the side effects of prolonged medical treatments.
And the damage itself, it can be caused by.
It can be mediated by autoantibodies, antibodies against self, or by self-reactive T cells, or often a combination of both.
Which brings us right back to Paul Ehrlich and his horror autotoxicus. For decades, the very idea of self-attack was considered, well, almost impossible, wasn't it? Who finally broke through that skepticism.
Yeah, that credit largely goes to Dr. Deborah Doniach and her colleagues in London. Back in 1957, she theorized, and then crucially demonstrated, that serum antibodies from patients with Hashimoto's thyroiditis actually reacted against normal thyroid components.
That must have been groundbreaking.
It really was. It was a pivotal moment, the first organ-specific autoimmune disease to be properly characterized. It finally shattered the scientific skepticism around the possibility of self-reactivity. Donich's work also helped identify an autoimmune factor involved in what was then called juvenile diabetes, now known as type 1 diabetes, another organ-specific autoimmune disease.
And today, sadly, diseases like rheumatoid arthritis, multiple sclerosis, lupus, they're all too common examples of what happens when tolerance fails.
Exactly. And we generally categorize them in a couple of ways, either as organ-specific, affecting primarily a single organ, or systemic, impacting multiple systems throughout the body, and also by whether antibodies, humoral immunity, or T cells, cell-mediated immunity, are the primary drivers of the damage.
Okay, let's unpack some specific examples then, starting with the organ-specific ones. What's happening in Hashimoto's thyroiditis?
Right. In Hashimoto's thyroiditis, individuals produce both autoantibodies and sensitized Th1 cells that are specific for thyroid antigens, particularly proteins called thyroglobulin and thyroid peroxidase.
Okay.
This disease is strikingly more common in women. The female-to-male ratio could be as high as 19-1, often striking in middle age.
19-1. Wow.
Yeah. And these antibodies, they interfere with the thyroid's ability to take up iodine, which is essential for making thyroid hormones. This leads to decreased thyroid function, or hypothyroidism.
And the T cells.
The sensitized Th1 cells drive what's called a delayed type hypersensitivity, or DTH response within the thyroid gland. This causes an intense infiltration of the thyroid by lymphocytes, macrophages, and plasma cells. They can even organize themselves into structures that look like lymph nodes, called lymphocytic follicles or germinal centers, essentially creating tertiary lymphoid organs within the thyroid.
So inflammation and damage.
Right. This inflammatory attack causes a goiter, that visible enlargement of the thyroid gland, and leads to symptoms like fatigue, lethargy, unexplained weight gain. Thankfully, it's often managed pretty effectively with daily thyroxine replacement therapy, just giving back the missing hormone.
And what about type 1 diabetes, T1D? That's another major organ-specific one and seems to be increasing.
Yes, type 1 diabetes, T1D, affects nearly 2 in 1,000 children here in the U.S., and that incidence has roughly doubled in the last 20 years or so, primarily affecting youth under 14.
And what's the target?
T1D is caused by an autoimmune attack against the insulin-producing beta cells, which are found in clusters called islets scattered throughout the pancreas.
Right, the islets of Langerhans.
Exactly. This attack involves cytotoxic T lymphocytes, CTLs, and activated macrophages. The process is often referred to as insulitis. This initiates a cell-mediated DTH response within the islets.
So T cells killing the beta cells.
Yes, and the resulting cytokine release from these T cells and macrophages, plus beta-glucan enzymes from the macrophages, directly destroys the beta cells. Autoantibodies, specific for beta cell antigens, are also produced, and they might contribute by facilitating complement lysis or maybe antibody-dependent cell-mediated cytotoxicity, ADCC.
And the consequences?
Decreased insulin production. This leads to increased blood glucose levels, serious metabolic problems like ketoacidosis, increased urine production, and in the longer term, it can cause complications like atherosclerotic vascular lesions, kidney failure, blindness. Untreated, it can certainly be fatal.
So daily insulin injections help manage it, but it's not a cure. Are there promising new treatments or good animal models for T1D?
Right, daily insulin is essential for survival, but it just can't replicate the body's own precise metabolically regulated insulin release. However, novel transplantation approaches, like transferring pancreatic islet cells themselves, are showing real promise, at least in mice, and if they can be translated clinically, they could offer a cure.
And animal models?
One of the best studied is the non-obese diabetic NOD mouse. These mice spontaneously develop diabetes that looks remarkably similar to human T1D. They get lymphocytic infiltration of the islets, beta cell destruction, and it's strongly associated with certain MHC alleles, just like in humans.
Okay.
Bone marrow transfer experiments in NOD mice showed the disease is clearly mediated by bone marrow-derived cells, presumably immune cells. And interestingly, remember the gut connection.
Yeah.
NOD mice housed in germ-free environments actually show a higher incidence of diabetes compared to those in regular housing.
So having gut microbes seems protective.
It suggests that yes, that a diverse gut flora might help block or delay autoimmune disease development. And human studies have also linked gut flora perturbations to T1D. Genome-wide scans have identified many genetic loci associated with T1D susceptibility in humans, including at least one member of the TNF receptor family. So it's complex, involving genes and environment, including our microbes.
Another well-known organ-specific autoimmune disease is myasthenia gravis. I understand that's a classic example of antibodies actually blocking normal function.
That's exactly right. Myasthenia gravis is really the quintessential example of an autoimmune disease mediated by blocking antibodies. Patients produce autoantibodies that bind to and physically block the acetylcholine receptors, AChRs, on the motor end plates of muscles.
So they block the signal from nerve to muscle.
Precisely. They prevent the normal binding of acetylcholine, the neurotransmitter. And not only that, the antibody binding also induces complement-mediated lysis of the muscle cells at the junction, leading to destruction of the AChR-bearing cells over time.
And the main symptom reflects that muscle weakness.
Yes, the primary symptom is a progressive weakening of the skeletal muscles. It often starts subtly, maybe with drooping eyelids or difficulty retracting the corners of the mouth, like smiling. Without treatment, it can progress to severe impairment in eating, swallowing, even movement.
How is it treated, and what have we learned from animal models there?
Treatment for myasthenia gravis aims to counteract the block. One way is to increase acetylcholine levels at the junction, for example, by using drugs that inhibit the enzyme that breaks down acetylcholinesterase inhibitors.
Okay.
Other approaches aim to decrease antibody production using corticosteroids or other immunosuppressants, or, in severe cases, actually remove the antibodies from the blood via plasmapheresis.
And the animal model.
There was a really serendipitous discovery back in 1973 that gave us one of the first good animal models, called experimental autoimmune myasthenia gravis, EAMG. Researchers immunized rabbits with purified ACRs from electric eels.
Electric eels. Okay.
Yeah. And unexpectedly, the rabbits suddenly became paralyzed. It turned out they developed antibodies that cross-reacted with their own rabbit AChRs, blocking muscle stimulation. This animal model quickly led to the discovery that autoantibodies to the AChR were also the cause of myasthenia gravis in humans.
Okay. So, moving from diseases that target just one organ, like the thyroid or the neuromuscular junction, to those affecting the whole body, we encounter systemic autoimmune diseases where the immune system seems to lose its compass on a much grander scale. What does that look like?
Right. In systemic autoimmune diseases, the autoreactive cells either target antigens that are found in many different tissues or organs throughout the body, or maybe there's just a widespread disruption in tolerance regulation itself, meaning anti-self cells aren't held in check properly anywhere. This leads to widespread tissue damage driven by cell-mediated immunity or autoantibodies or the accumulation of immune complexes or often combinations of these.
And one of the most recognized systemic autoimmune diseases is systemic lupus erythematosus, SLE, or just lupus.
SLE is a prime example. And like many autoimmune syndromes, it's significantly more common in women than men, about a 9-to-1 ratio. Symptoms typically show up between ages 20 and 40, and it occurs more frequently in African American and Hispanic women, though the reasons for that aren't fully understood yet.
Is there a genetic link?
Yes, there's a strong genetic component. Identical twins have up to a 60% chance of both developing SLE if one twin has it, though, interestingly, it's only about 2% for other close relatives, which tells you that other factors, non-genetic factors, must also be heavily involved.
And what's the immune system attacking in lupus?
It's quite remarkable, actually. Individuals with SLE can produce autoantibodies to a huge array of common cellular components. Things like DNA, histones, the proteins DNA wraps around, clotting factors, even blood cells themselves, red blood cells, platelets, white blood cells.
Wow, that's a massive range of targets. What are the typical symptoms and how does the damage actually happen?
Symptoms can be quite varied, but often include fever, weakness, arthritis, kidney dysfunction, and skin rashes, including that characteristic butterfly rash across the nose and cheeks.
Okay.
When autoantibodies target red blood cells or platelets, it can lead to their destruction through complement-mediated lysis, causing hemolytic anemia or thrombocytopenia. But a critical mechanism of damage in SLE involves immune complexes.
Immune complexes, antibodies bound to antigens.
Exactly. Autoantibodies bind to various nuclear antigens released from damaged cells, forming these complexes. These immune complexes then get deposited along the walls of small blood vessels, especially in the kidneys, skin, and joints.
And that deposition causes problems.
Big problems. It triggers a type 3 hypersensitivity reaction. The complexes activate the complement system, generating membrane attack complexes and complement fragments like C3a and C5a, which are highly inflammatory. This damages the blood vessel walls, resulting in vasculitis, inflammation of the blood vessels, and glomerulonephritis, inflammation of the kidney's filtering units. In severe cases, this can lead to neutrophil aggregation, low neutrophil counts, neutropenia, and blockages in small blood vessels, causing widespread tissue damage.
How is lupus usually diagnosed?
Diagnosis often involves detecting anti-nuclear antibodies, or ANA, in the patient's blood. This is typically done using indirect immunofluorescence, where the patient's serum is incubated with cells, and if ANAs are present, they bind to the cell nuclei, producing a characteristic staining pattern that can be seen under a microscope.
Is there an animal model for lupus?
Yes, a very good one actually. The New Zealand mouse, specifically the NZBWF1 hybrid strain. These mice spontaneously develop a severe syndrome that closely resembles human SLE, including the autoantibody production and kidney damage. And interestingly, just like in humans, the disease incidence is much higher in female mice.
Okay. Another devastating systemic disease is multiple sclerosis, MS, affecting the nervous system.
Yes, MS is the most common cause of neurologic disability, particularly in Western countries. And again, it affects women much more frequently than men, about 3 times more often now. That ratio has actually increased over the last couple of decades, which suggests recent increases in MS incidence might have been primarily in women.
Interesting. And the age range?
Like SLE, it often develops in young to middle adulthood, typically between 20 and 40 years old.
And what's the immune attack directed against in MS?
MS is primarily driven by autoreactive T cells, particularly CD4 plus T cells, and specifically Th17 cells, and the cytokine they secrete, IL-17, are considered hallmarks of the condition. These T cells, along with other immune cells they recruit, lead to inflammatory foci, or lesions, along the myelin sheath of nerve fibers in the brain and spinal cord.
Myelin, the insulation around nerves.
Exactly. Since myelin insulates nerve fibers and allows for rapid signal transmission, its breakdown leads to progressive neurologic dysfunction. Symptoms can range widely, from numbness and tingling to paralysis or vision loss. The most common form of MS is relapsing and remitting, where symptoms flare up and then subside, but a chronic progressive form also exists.
What factors contribute to someone developing MS? It sounds complex.
It is very complex. There are both strong genetic and environmental associations. Certain MHC class II alleles, particularly HLA-DRB1 variants, have long been linked to MS risk. But genome-wide studies have now identified many other genetic loci involved, often related to immune function.
So genes play a role, but it's not purely genetic.
Definitely not. Genetics influence susceptibility, but they aren't deterministic. For example, if one identical twin has MS, the other twin only has about a 1 in 4 chance of developing it, compared to maybe 1 in 1,000 for the general population. This clearly points to significant environmental factors.
Like what?
Well, epidemiological studies show MS is more common in the northern hemisphere, further from the equator. And if someone relocates from a low incidence region to a high incidence region early in life, their risk increases. This suggests environmental factors encountered early on are important. Things like diet, smoking, obesity have been implicated. And even exposure to sunlight, which is linked to vitamin D levels, and vitamin D is known to be an immune modulator.
Interesting. What about infections?
Infection with the Epstein-Barr virus, EBV, has long been cited as a possible trigger or contributing factor, perhaps explaining some of the geographic gradient. And the hygiene hypothesis, the idea that lack of early life exposure to microbes might predispose to immune dysregulation, may also play a role.
Is there an animal model for MS?
Yes, a very important one called experimental autoimmune encephalomyelitis, EAE. It's usually induced in rodents by immunizing them with components of myelin, like myelin basic protein, MBP, or proteolipid protein, PLP. These animals develop CNS inflammation, demyelination, and paralysis that mimics many aspects of human MS, including the relapsing remitting course in some models. EAE has been absolutely crucial for understanding the disease mechanisms and testing potential therapies.
And that diet and microbiome connection we discussed earlier, does that apply to MS and EAE too?
Absolutely. Recent work, particularly using the EAE model, really highlights the increasingly appreciated role of diet and the gut microbiome in MS susceptibility and progression. Remember those germ-free mice? They're less susceptible to EAE. And specific gut commensal bacteria have been shown to either exacerbate or attenuate the disease in mice and human MS patients. They also show disruptions in their gut flora, especially during flare-ups. So this is a really important area for testing causal hypotheses and maybe developing new treatments focused on the gut.
Okay, finally, let's touch on rheumatoid arthritis, RA. Another common systemic autoimmune disease known especially for its impact on the joints.
Right. RA is a fairly common autoimmune disorder, usually diagnosed between ages 40 and 60, and again, significantly more frequent in women. It also has a strong genetic susceptibility component, with the HLA-DRB1 locus again implicated, along with many other non-MHC genes.
And the main symptom is joint inflammation.
That's the major symptom, yes. Chronic inflammation of the joints, leading to pain, swelling, stiffness, and eventually destruction of cartilage and bone. But RA can also affect other systems, including hematologic, cardiovascular, and respiratory systems.
What are the key immune players in RA?
Two things are particularly characteristic. First, most RA patients produce antibodies against citrullinated protein antigens. Citrullination is a modification where an arginine amino acid residue is converted to citrulline. Why antibodies form against these modified proteins is a key question.
Okay, anti-citrullinated protein antibodies, or ACPAs, what's the second thing?
The second is the production of autoantibodies called rheumatoid factors, RFs. These are quite unusual. They are antibodies that are specific for the FC region of other antibodies, specifically IgG. Essentially, antibodies against antibodies. The classic RF is an IgM antibody that binds to IgG, but RFs of other isotypes like IgG or IgA can also exist.
So these rheumatoid factors are central to the joint damage. How does that work?
Yes, they play a significant role. When RFs bind to normal circulating IgG, they form large immune complexes. These complexes get deposited in the joints, particularly in the synovial fluid and lining.
Ah, immune complex deposition again, like in lupus.
Exactly. These deposited complexes activate the complement cascade, leading to a type 3 hypersensitivity reaction. This drives chronic inflammation within the joint, attracting inflammatory cells, releasing damaging enzymes and cytokines, and ultimately leading to the progressive destruction of the joint tissues.
Are there environmental links for RA too?
Yes. Like SLE and MS, RA has recently been connected to our body flora. Specifically, it's associated with gum disease, periodontitis, caused by bacteria like Porphyromonas gingivalis. Smoking is also a major environmental risk factor.
How might gum disease or smoking trigger RA?
Well, one leading hypothesis is that inflammation in the gums or in the lungs due to smoking might increase the process of protein citrullonation in those mucosal tissues. This could then trigger the production of those anti-citrullinated protein antibodies, ACPAs, in individuals who are genetically susceptible. So the initial trigger might be local, but the autoimmune response becomes systemic and targets the joints.
Okay, so beyond diseases targeting specific organs or having these widespread systemic effects, sometimes the core issue isn't what the immune system targets, but a fundamental flaw in its ability to regulate itself, often stemming from just one single genetic glitch.
Precisely. This represents a relatively newer category of autoimmune disease, linked directly to genetic disruptions in fundamental immune regulation. The two classic examples here are autoimmune polyendocrine syndrome, type 1, APS1, and immune dysregulation, polyendocriopathy, enteropathy, X-linked IPEX syndrome.
APS1 and IPEX, they sound serious.
They are very serious, and despite affecting different genes, they share many features. Both are monogenic disorders, meaning they're caused by a mutation in just a single gene. They impact multiple organs, particularly endocrine glands, and they display a similar, rather paradoxical range of immunopathologies, including severe autoimmunity and aspects of primary immune deficiency.
How can one gene cause both too much immune response, like autoimmunity, and also not enough, like immune deficiency? That seems contradictory.
It does seem paradoxical, but it all comes back to that concept of balance and a profound disruption to homeostatic immune regulation, a fundamental break in self-tolerance.
So what are the genes involved?
APS1 is caused by mutations in the AIR gene. Remember AIR?
Yeah, the autoimmune regulator in the thymus, crucial for central tolerance.
Exactly. AIR ensures that those tissue-specific antigens get expressed in the thymus during T-cell development. This leads to both the elimination of T cells that recognize these self-antigens too strongly, and also the positive selection or engagement of regulatory T cells that recognize them. So if AIR is mutated, that whole central training process is fundamentally flawed.
Okay. And IPEX syndrome.
IPEX syndrome is caused by mutations in the FOXP3 gene. And FOXP3, as we discussed, is the absolute master transcriptional regulator for regulatory T cells, both the Tregs generated in the thymus and the Tregs induced in the periphery.
Ah, so both AIR and FOXP3 defects hit Treg function hard.
Precisely. The common denominator for both APS1 and IPEX is a critical loss of regulatory T cell function and a profound disruption to self-tolerance. This leads to widespread dysregulated immune activity. So it's not really about the immune system having too much of one thing and too little of another, but rather a fundamental flaw in its ability to control itself, which manifests as both autoimmunity and aspects of immunodeficiency because that crucial regulatory arm is missing or broken.
Okay, that makes sense. So zooming out again, what are the general triggers that tip this delicate balance toward developing an autoimmune disease? It sounds like it's rarely just one single thing.
You're absolutely right. It's almost always a complex interplay of intrinsic factors, mainly genetic predisposition, and extrinsic or environmental factors. Plus, as we've seen, sex plays a major role, and there's probably an element of random chance or stochastic events involved too.
Let's start with the genes. You mentioned MHC or HLA in humans. That's a big one.
It's the top genetic contributor. Yes, primarily because of its central role in presenting antigens to T cells. Specific MHC class 1 and class 2 alleles, or sometimes mutations within them, are commonly associated with particular autoimmune diseases.
Is there a really strong example?
The strongest known association is between ankylosing spondylitis, AS, that's an inflammatory disease primarily affecting the vertebral joints, and the HLA-B27 allele. Having HLA-B27 increases an individual's risk of developing AS by an astonishing 90-fold compared to someone without it.
90-fold. Does everyone with HLA-B27 get AS?
No, and that's crucial. Most individuals with HLA-B27 will not develop AS. It tells us that while HLA-B27 confers significant susceptibility, other genetic or environmental factors must also be involved to trigger the disease. Interestingly, AS is one of the few autoimmune diseases that's actually more common in men than women, and it's also been linked to genes involved in the IL-23, IL-17 pathway, pointing towards Th17 cell function.
So it's not just MHC genes. What about other genes involved in the immune response itself?
Right. Beyond MHC, many other non-MHC immune genes are associated with autoimmune disease risk. We've already highlighted the critical examples of AIR and FOXP3 mutations leading to severe systemic conditions. But there are many others. Genes for cytokines and their receptors, components of the antigen processing and presentation machinery, adhesion molecules that help cells interact, and importantly, costimulatory or inhibitory receptors like CTLA4 or PTPN22 variations in these genes have all been linked to susceptibility to one or more autoimmune diseases.
PTPN22, what does that do?
PTPN22 encodes a tyrosine phosphatase, an enzyme involved in regulating signaling pathways. A particular variant of PTPN22 has been linked to increased risk for several autoimmune diseases, including type 1 diabetes, rheumatoid arthritis, and SLE. It's thought that this variant might slightly dampen T-cell receptor signaling during T-cell selection in the thymus, paradoxically making it easier for weakly autoreactive T-cells to escape deletion and potentially cause trouble later.
So multiple genes can contribute, often interacting with each other and the environment.
Exactly. In many cases, it's likely an accumulation of several susceptibility gene variants combined with the right environmental triggers that finally pushes someone over the threshold into clinical autoimmune disease.
Okay, let's talk more about those environmental factors. What pulls the trigger?
Environmental factors seem to play a universal role in converting genetic predisposition into actual disease. Beyond maybe the obvious suspects like smoking, obesity, or certain chemical exposures, diet and our own mucosal flora, especially in the gut, have increasingly taken center stage.
We talked about the geographic links for MS.
Right. Autoimmune syndromes are often more common in certain geographic locations or climates, suggesting links to environmental and lifestyle factors prevalent there. And the gut microbes, sometimes called our old friends, they're in constant communication with immune cells in the intestinal lining and elsewhere. This crosstalk isn't just local. It influences systemic immunity, helping to regulate peripheral tolerance and potentially suppress autoimmune disease developing in distant sites.
And the germ-free animal studies support this.
Yes. Animals raised in sterile, germ-free environments often show heightened susceptibility to various autoimmune diseases compared to their conventionally raised microbe-laden counterparts. It really emphasizes this connection. This interplay between our gut microbiome and our overall immune health is a truly fascinating and rapidly evolving area.
Infections have also been strongly implicated, correct? How might getting an infection lead to an autoimmune disease later on?
Yes, infections are definitely thought to be major environmental triggers for autoimmunity, and they can do it through several potential mechanisms. First, simple tissue damage during an infection can lead to the release of normally sequestered self-antigens.
Antigens that were hidden away before.
Exactly.
Yeah.
If these newly exposed self-antigens are then presented to the immune system in a context of inflammation and danger signals from the infection, an autoimmune response can be triggered against that tissue. For example, the release of heart muscle antigens after a heart attack can sometimes lead to autoantibodies targeting healthy heart muscle.
Okay. What else?
A second major mechanism is molecular mimicry. This is the idea that some pathogens might express protein epitopes, small parts of proteins that happen to resemble components of our own self-tissues, either in their 3D shape or their amino acid sequence.
So the pathogen looks like self...
Kind of, yeah. When the immune system mounts a response against the pathogen, it might inadvertently activate T or B cells whose receptors cross-react with the similar-looking self-component. If this happens in a pro-inflammatory environment driven by the infection, it could effectively bypass normal tolerance mechanisms and initiate autoimmunity.
Is there a classic example of molecular mimicry?
The classic example is rheumatic fever, which can develop a few weeks after a group A streptococcus infection, like strep throat. Antibodies generated against certain streptococcal antigens have been shown to cross-react with proteins in the heart muscle, leading to carditis, a type 2 hypersensitivity reaction damaging the heart. Studies have even suggested that a small percentage, maybe over 3%, of antibodies raised against common viruses might bind to normal host tissues, indicating that this kind of molecular similarity might be fairly common. And again, an individual's MHC haplotype can influence whether these cross-reactive peptides are presented effectively, affecting susceptibility.
Okay, sequestered antigens, molecular mimicry. Any other ways infections can trigger autoimmunity?
A third possibility is polyclonal activation. Some infectious agents, like certain gram-negative bacteria with LPS, or viruses like cytomegalovirus, CMV, or Epstein-Barr virus, EBV, can act as potent B-cell mitogens. They can cause widespread, nonspecific proliferation of many different B-cell clones, potentially including some that are autoreactive. This can lead to the appearance of autoantibodies even without specific T-cell help. If B-cells reactive to self-antigens get activated this way, autoantibodies can appear and potentially contribute to disease. So you can see maintaining control over autoreactivity is this constant daily battle influenced by our genes, our environment, our infections, and probably just random chance.
And we've touched on how women are disproportionately affected by most autoimmune diseases. Why is that? What are the key sex differences that might explain this?
Yeah, the statistics are really stark. Something like 80% of all autoimmune patients in the U.S. are women. We mentioned the ratio for Hashimoto's can be 19 to 1. For lupus, it's around 9 to 1. While the reasons aren't completely understood, recent advances are helping scientists form testable hypotheses.
What are some of those hypotheses?
Well, generally speaking, females tend to mount more vigorous innate and adaptive immune responses than males. They often clear infections faster, produce higher antibody titers after vaccination, for instance. But this enhanced immunity in females seems to come with a trade-off. Paradoxically, males are generally more prone to infections and maybe certain cancers, but they are less likely to be diagnosed with autoimmune disorders.
So, a stronger immune system might be more prone to mistakes, like attacking self. What role do sex hormones play?
That's the prevailing view, yes. Sex hormones likely account for at least part of this difference. Estrogens, which are obviously higher in females, are generally associated with enhancing immunity. Androgens, like testosterone, which are higher in males, are generally associated with suppressing immunity.
Is there evidence for that?
Yes, quite a bit. For example, SLE flares often occur during pregnancy, which is a very high estrogen state. And remission rates for SLE tend to increase after menopause when estrogen levels drop significantly. In animal models, if you castrate male mice, their immune responses start to look more like female responses, including increased susceptibility to autoimmune diseases. And recently, work has even suggested that sex hormones might affect the composition of the gut microflora, which, as we know, influences systemic immunity. For example, transferring male-specific microbiota into female NOD mice actually protected them from developing diabetes.
Wow. This also connects to the idea I've heard that women's increased risk might somehow be a byproduct of their evolutionary role as childbearers. How does pregnancy itself factor in?
Pregnancy definitely gives us important clues. It's absolutely critical for the mother's immune system to tolerate the fetus, which is essentially a semi-allograft, half-foreign genetically. This requires significant modifications to the female immune system during gestation.
How does it change?
While females normally might tend towards more Th1-like pro-inflammatory responses, during pregnancy, there's a notable shift towards more Th2-like anti-inflammatory responses. This is thought to be promoted by the high levels of pregnancy-associated sex steroids, creating an overall anti-inflammatory environment conducive to fetal survival.
And this affects autoimmune diseases.
It seems to. Diseases that are thought to be exacerbated by Th2-like responses, such as SLE, can actually worsen during pregnancy, whereas inflammatory diseases, usually driven by Th1 or Th17 cells, like rheumatoid arthritis or MS, are often ameliorated. They get better during pregnancy.
That's fascinating. What about the longer-term impact of pregnancy on the mother's immune system? Anything lasting?
Yes, another really fascinating effect of pregnancy is bidirectional microchimerism. This is the exchange of small numbers of cells between the mother and the fetus in both directions. And these exchanged cells can persist in the mother or the child for decades after the pregnancy.
Decades, wow. Does that affect immunity?
There's interesting, though still somewhat limited, evidence suggesting that these long-lived fetal cells in the mother, or maternal cells in the offspring, might influence immune regulatory balance later in life, and thus potentially affect the development of autoimmune disease. For instance, some studies have found that women who develop certain autoimmune diseases postpartum, like MS, Hashimoto's, or Graves' disease, show higher levels of fetal microchimerism compared to healthy controls. This raises a really important question. What other long-term immune effects does this cellular exchange have on both the mother and the child throughout their lives? We're still figuring that out.
Okay. One last piece on autoimmunity before we move on. Which specific T helper cell types are thought to be the main bad actors when tolerance breaks down?
Well, CD4 plus T cells, more so than CD8 plus T cells, have been consistently linked to initiating and driving most autoimmune diseases. Historically, a lot of the early data pointed towards autoreactive Th1 cells and the IFN gamma they produce being the primary culprits. For example, IFN gamma levels in the CNS of mice with EAE correlated pretty well with disease severity, and treating human MS patients with IFN gamma actually exacerbated their disease.
Okay, so Th1 and IFN gamma seemed key.
They did. But then some conflicting results emerged. For instance, genetically eliminating IFN gamma in mice sometimes unexpectedly worsened EAE in certain models. This prompted researchers to look more closely at other T helper cell types, and that's when Th17 cells really came into the spotlight as important players in autoimmunity.
Th17 cells, how did they become implicated?
Remember, cytokines like IL-12 and IL-23, usually produced by antigen-presenting cells, help steer T cell development toward either the Th1 or the Th17 pathway. Studies started showing that if you blocked key signals needed to generate or sustain these Th17 cells, particularly signals involving IL-23, the mice were protected from developing EAE, or collagen-induced arthritis, model for RA. Likewise, mice genetically lacking the main Th17 cytokine, IL-17A, were found to be much less susceptible to developing these autoimmune models. And importantly, in human patients with diseases like MS, RA, psoriasis, and SLE, elevated levels of IL-17 and or IL-23 have been found at the sites of inflammation and sometimes in their serum.
So the evidence points strongly to Th17 cells now.
Yes. Collectively, these findings provide very strong support for the idea that Th17 cells are important drivers, maybe even central drivers, of multiple autoimmune diseases. And not surprisingly, this has led directly to the development of new immunotherapies, specifically targeting the IL-23 Th17 pathway, some of which are now used clinically.
Okay, so now that we've journeyed through the intricate dance of immune tolerance and the often tragic breakdown that is autoimmunity, let's turn to a field where controlling that very dance is, quite literally, a matter of life and death: transplantation. The story here, from ancient attempts to modern miracles, is truly remarkable, isn't it?
It really is. The earliest accounts we have of tissue transplantation actually date way back to India in the 5th century BC. The legendary surgeon Sushruta described reconstructing noses using skin transferred from another site on the same individual.
Ah, so an autograft, using your own tissue.
Exactly. And today we know that's the type of transplant most likely to succeed because there's no foreignness, no immune barrier. It wasn't really until the early 20th century, 1908, that Alexis Carrel conducted the first systematic studies transplanting kidneys in cats. He showed that a transplanted organ could actually function in a new recipient, with some of his cat kidneys producing urine for up to 25 days. A huge step.
And then, of course, the pivotal moment came with human-to-human transplantation.
Yes, in 1954, Joseph Murray and his team in Boston achieved the first truly successful long-term organ transplant: a kidney between identical twins. Because they were genetically identical, there was no immune rejection.
Right, but the real challenge was transplanting between unrelated people.
Precisely. Overcoming the formidable barrier of the MHC molecules and the resulting alloreactivity, that immune response to foreign MHC, that was the key. And the true leap forward there came in 1961. And this is where the story gets really interesting. Dr. Samuel Counts, who was a pioneering African-American transplant surgeon at Stanford at the time, led a team that performed the first successful non-twin living-related human kidney transplant from a mother to her daughter.
Wow, that must have been groundbreaking.
Absolutely. Their pioneering work, which combined early immune suppressants with new techniques for perfusing and preserving the kidney, truly heralded a new era. It allowed physicians to start imagining transplantation not just as an experiment, but as a viable cure for organ failure.
So today, after all those breakthroughs and decades of refinement, where do we stand with transplantation?
Well, today, the transfer of organs and tissues between non-identical individuals' allografts is performed with ever-increasing frequency and success. In 2016 alone, for instance, over 30,000 solid organ transplants were performed just in the United States, plus over 46,000 corneal tissue grafts.
That's a lot.
It is. And clinical outcomes, especially short-term survival, have improved considerably, largely thanks to better immunosuppressive drugs. However, major obstacles definitely persist. Those immunosuppressive drugs, while essential, come with significant long-term medical problems. And even with better short-term success, chronic rejection remains a really difficult, lingering problem. It often leads to the eventual failure of the graft, necessitating second or third transplants, which, of course, only exacerbates the already severe shortage of donor organs.
And this brings us to that stark reality. Despite all the incredible medical advances, there just aren't enough organs to go around for everyone who needs one.
That's the unfortunate, dire truth. Demand for donor organs and tissues continually outstrips supply, by almost 4 to 1 in many cases. The American Transplant Foundation estimates that about 20 people die every single day in the U.S. just waiting for a transplant.
20 people a day? That's staggering.
It is. As of March 2018, there were nearly 115,000 individuals on waiting lists in the U.S., with kidney patients often facing the longest waits, sometimes 3 to 5 years, or even more. And this severe shortfall has forced the medical community to seriously consider alternatives, including xenotransplantation.
Xenotransplantation, using animal organs to address the human organ deficit. That sounds like something out of science fiction, but it's real.
It's becoming science fact, yes. There were some early clinical attempts using non-human primates, chimpanzees, baboons as donors. They showed some very limited short-term success, like a baboon liver functioning for maybe 70 days, or a chimp kidney for 9 months.
But primates aren't ideal.
No. Their close phylogenetic relationship to humans actually carries an elevated risk of transferring dangerous pathogenic viruses. Plus, there are major practical and ethical concerns with using primates. So, attention shifted towards pigs.
Pigs? Why pigs?
Pigs have been under serious consideration for many years now. They have several advantages. They breed rapidly, have large litters, they can be housed in controlled, pathogen-free environments to reduce infection risk, and their organs are anatomically and physiologically quite similar in size and function to human organs. In fact, cardiac valves from pigs have been successfully used in human heart surgery for years.
But I understand there's a massive immune hurdle with pig organs, something called hyperacute rejection. How does that work?
That's the major immediate difficulty, yes. If you were to implant a standard pig organ into a human, it would likely fail within minutes to hours due to hyperacute rejection.
Minutes to hours? Wow. Why so fast?
It's an antibody-mediated response. It's caused primarily by the presence of a specific sugar molecule, a disaccharide called galactose alpha-1-parent-3-galactose, or IGAL for short, which is expressed on the surface of pig cells, but not human cells.
IGAL? And we have antibodies against it.
Pretty much all humans do, yes. We have pre-existing antibodies against IGAL because we get exposed to this sugar, or very similar structures, on many common bacteria, including the ones living in our own gut. So when these pre-existing human antibodies encounter the IGAL on the pig organ's blood vessel lining, they bind immediately.
And that triggers rejection.
Very rapidly. It triggers the complement cascade, leading to inflammation, blood clotting, and destruction of the pig endothelial cells. The blood vessels in the graft get blocked and the organ dies from lack of blood flow. The problem is also intensified because pig cells lack some of the key human complement regulatory proteins, like AF and MCP, that normally help put the brakes on complement activation on our own cells. So the attack is uncontrolled.
Okay, that sounds like a showstopper. How can this massive hurdle possibly be overcome?
Well, trying to deplete the human and TK-cal antibodies or block complement hasn't been very successful long term. A much more elegant solution has involved genetic engineering of the pigs themselves.
Engineering the pigs? Wow.
Scientists have successfully created pigs with a gene knockout for the enzyme alpha-1 galactosyl transferase that's responsible for adding that IGAL sugar onto proteins. These are called the GalTKO pigs for galactosyl transferase knockout. They simply lack the IGAL antigen on their cells.
So no target for the pre-existing antibodies. Does it work?
It helps tremendously. Kenji Kawaki and colleagues, for instance, transplanted hearts from these GalTKO pigs into baboons. They achieved a mean survival time of 92 days, with one heart impressively surviving for 179 days. Another group, led by Kazuhiko Yamada, transplanted GalTKO pig kidneys into baboons and saw function for up to 83 days. They even tried simultaneous transplantation of the pig thymus, hoping to induce tolerance in the baboons. While not completely solving all rejection issues, these results with GalTK organs were incredibly promising and have spurred much further research into multi-gene modified pigs.
Are there other concerns with pig organs besides that initial hyperacute rejection?
Yes, another significant concern revolves around pig endogenous retroviruses, or PRVs. These are retroviral sequences integrated into the pig genome. There's a theoretical risk that these could be transferred to the human recipient and potentially cause new types of infections or even trigger a new epidemic, perhaps something like HIV. So there's ongoing work to develop PRV-free pig lines to mitigate this risk.
Okay. What about other futuristic approaches, like chimeric animals?
That's another cutting-edge area. Back in 2017, there were breakthroughs from 2 different research teams who successfully created chimeric or interspecies animals as a proof of concept. One group grew human cells within a developing pig embryo. Another grew viable mouse cells, including pancreas precursors, within a rat embryo.
Wow. So you could potentially grow a human organ inside another animal.
That's the long-term hope. These chimeric fetuses were only allowed to develop for a few weeks due to regulatory and ethical guidelines, but the approach is seen as a major potential advance for generating future organs or cells for transplants. Of course, there are still significant ethical and regulatory hurdles to overcome before this could ever become a clinical reality.
Okay. So, given all these challenges, whether using human or potentially animal organs, how do doctors try to find the perfect match, or at least a good enough one, between a specific donor and a recipient?
Finding the best possible match based on antigenic similarity is absolutely crucial, as it directly improves the chances of transplant success. We generally classify grafts into four main types based on the genetic relationship between donor and recipient.
Right. You mentioned autografts and xenografts. What are the others?
An autograft, as we said, is self-tissue transferred within the same individual. An isograft is tissue transferred between genetically identical individuals, like identical twins, or mice from the same highly inbred strain. An allograft is tissue transferred between genetically different members of the same species. This is the most common type of transplant in humans. And a xenograft, as we discussed, is tissue transferred between different species.
And the success rate varies dramatically.
Absolutely. Autografts and isografts are usually accepted without immune rejection because there's no genetic difference, no foreign antigens. Allografts, having genetic differences, will elicit an immune response and be rejected unless immunosuppression is used. And xenografts, with the greatest genetic disparity, face the most rapid and vigorous rejection. Tissues that share sufficient antigenic similarity to allow transfer without rejection are called histocompatible.
And what's the biggest factor determining histocompatibility or incompatibility?
By far, the MHC, the major histocompatibility complex, which we call HLA, human leukocyte antigen, in humans, is responsible for the most vigorous allograft rejection reactions. Remember, MHC genes are inherited as a set or haplotype from each parent.
Right.
This means that in outbred human populations, there's only about a 1 in 4 chance of finding a perfect MHC identity between any two siblings. And a parent-to-child graft, while always a 50% match for one parental haplotype, is still mismatched for the other parent's alleles. So finding a good HLA match is key.
So, to predict and improve success, what tests are done before a transplant?
There are 3 critical pre-transplantation tests performed routinely. First, ABO blood group matching. Second, MHC matching, also called tissue typing or HLA typing. And third, cross-matching.
Okay, let's take those one by one. Why is blood type ABO matching so critical even for an organ transplant, not just a blood transfusion?
It's absolutely critical because ABO blood group incompatibility between donor and recipient usually causes the most intense and rapid form of rejection, that hyperacute rejection we talked about.
Because the antigens are on the organ cells, too.
Exactly. Blood group antigens aren't just on red blood cells, they're also expressed on epithelial cells, and crucially on the endothelial cells that line the blood vessels within the transplanted organ. If the recipient has pre-existing antibodies against any of the donor's blood group antigens, like a type B person having anti-A antibodies, those antibodies will immediately bind to the graft endothelium, trigger complement, and destroy the organ. So ABO compatibility is almost always the first non-negotiable requirement.
Okay, ABO match first. Then what about MHC matching or tissue typing? How precise does that need to be?
The level of precision needed or achievable for MHC matching depends a bit on the type of transplant and the urgency. For living donations or transplants where MHC matching is particularly critical, like bone marrow transplants, parents or siblings are usually the preferred donors because of the higher likelihood of finding a partial or even full HLA match.
But for organs like hearts or lungs?
Right. For solid organs, especially urgent ones like heart or lungs that can only survive for a few hours outside the body, rigorous, high-resolution MHC matching often isn't feasible due to the time constraints. The priority is getting a healthy organ transplanted quickly, often requiring semi-local donation networks. However, for bone marrow transplants, where the donor's immune cells need to function properly within the recipient's body, which requires recognizing the recipient's MHC, achieving the best possible HLA match is absolutely crucial for success and minimizing complications like GVHD.
How is HLA typing actually done now?
It used to be done using serologic methods, using antibodies to detect HLA proteins, but now molecular assays are much more common. These use techniques like PCR with sequence-specific primers or even DNA sequencing to determine the specific HLA alleles the donor and recipient carry. Molecular methods offer much higher resolution and specificity.
Even with a perfect MHC match, can rejection still happen?
Yes. Even with identical MHC matching, differences at other genetic locations, called minor histocompatibility loci, can still elicit an immune response and cause rejection, although it's typically slower and less vigorous than MHC mismatched rejection. This means some level of immunosuppression is almost always required even for MHC identical allografts. except between identical twins.
Okay, and what about that final, you said maybe most important test,
No-
the cross match?
Yes, the cross-match is arguably the most critical compatibility test performed just before a solid organ transplant. Its purpose is to detect any preformed recipient antibodies that react specifically against the potential donor's alloantigens, primarily their HLA molecules.
And if the cross match is positive,
A positive cross-match means the recipient already has antibodies circulating that will attack that specific donor's tissue. Transplanting in this situation almost guarantees rapid hyperacute rejection. So a positive cross-match is generally an absolute contraindication to proceeding with that particular donor-recipient pair.
where do these performed anti-HLA antibodies come from?
They can arise from several sensitizing events. Previous blood transfusions, which contain white blood cells expressing HLA, previous pregnancies where the mother can become sensitized to paternal HLA antigens expressed by the fetus, exposure to certain infectious agents that might elicit cross-reactive antibodies, or, of course, a previous transplant that failed.
And the timing is important.
Absolutely. Because a recipient's antibody profile can change over time, the cross-match test is always performed shortly before the planned transplant surgery, using current samples from both donor and recipient. The most common method today is the Luminex assay, which uses tiny microbeads coated with specific known HLA proteins. The recipient's serum is incubated with these beads, and sophisticated detectors measure if antibodies bind to any specific HLA types, giving a detailed profile of the recipient's anti-HLA antibodies.
Is there data showing how important cross-matching is?
Oh yes, a seminal study back in 1969 clearly demonstrated its value. They found that about 80% of kidney transplant patients who had received a graft, despite having a positive cross-match, experienced almost immediate rejection. In contrast, only about 5% of patients with a negative cross-match experienced this type of early failure. It really underscores why this test is so crucial for preventing hyperacute rejection.
Okay. Now, thinking about the different organs themselves, are some organs generally easier to transplant than others? What are the specific challenges for different types?
Yes, absolutely. The frequency with which different organs are transplanted really depends on a combination of factors. Are there good alternative treatments available? How readily available are donor organs? How complex is the surgery? How long can the organ survive outside the body?
Which organs are most commonly transplanted?
Kidneys are by far the most commonly transplanted organ, making up about 75% of all solid organ transplants in the U.S.
Why kidneys so often?
Several reasons. Kidney failure is unfortunately increasingly common, often linked to diseases like diabetes and hypertension. Also, we have two kidneys, so healthy individuals can donate one as a living donor, which expands the donor pool significantly. The surgery itself, while complex, is relatively more straightforward than, say, heart or liver transplants. Kidneys also tolerate being outside the body, ex vivo, longer than hearts or lungs, allowing more time for transport and matching. And because kidney transplantation has been done for so long, there are well-established patient care protocols and effective immunosuppressive regimens.
What about bone marrow or hematopoietic stem cell, HSC transplants? Those are quite different, involving cells rather than a solid organ.
Indeed. Bone marrow BM, or hematopoietic stem cell HSC transfer, is primarily used to treat hematologic diseases, cancers like leukemia and lymphoma, or genetic disorders like severe combined immunodeficiency, SCID. The goal is to replace the patient's diseased hematopoietic system with a healthy one from a donor.
Is finding a donor hard?
Yes. Finding a suitably MHC-matched donor is also the biggest hurdle for BMHSC transplant. While bone marrow is technically a renewing resource, the pool of registered volunteer donors is limited, especially for individuals from ethnic minorities, who often have less common HLA types. Achieving a close HLA match is critically important here.
Why is the match so critical for bone marrow?
Because you're transplanting an entire immune system. The recipient is usually heavily immunosuppressed before the transplant to prevent rejection of the graft. But the opposite problem, graft versus host disease, GVHD, becomes a major challenge.
Graft versus host, the donor cells attack the recipient.
Exactly. The immunocompetent donor T cells within the graft recognize the recipient's tissues as foreign due to any remaining HLA mismatches or minor antigen differences, and mount an attack. GVHD can affect the skin, liver, gut, and other organs and can be very severe, even fatal. While techniques like depleting T cells from the donor marrow before infusion have helped reduce GVHD incidence and severity, it remains a major obstacle limiting the success and broader application of bone marrow transplantation.
And for vital organs like the heart and lung, where time is absolutely critical.
These are arguably the most dramatic forms of transplantation. Yeah. The biggest challenge for heart and lung transplants is their extremely short ex vivo viability. They can only survive for a few hours preserved on ice. This means the recipient must be ready, often kept alive by artificial means, and the donor organ needs to be transported very quickly.
Does that limit matching?
Yes, because of this urgency and the limited supply, rigorous HLA matching often isn't possible for heart and lung transplants. The priority is getting a suitable, healthy organ transplanted before it deteriorates. Despite this lack of precise matching, the 1-year survival rate for heart transplants, for example, has climbed to over 80% thanks to effective immunosuppression. And new technologies, like the heart-in-a-box systems that perfuse the organ with warm oxygenated fluid, are aiming to extend that viability window significantly, which might allow for better matching in the future.
What about other organs like the liver or the pancreas?
The liver presents unique technical challenges due to its complex circulatory network and role in metabolism. However, its large size and regenerative capacity mean a single donor liver can sometimes be split and transplanted into 2 recipients, often an adult and a child. Liver transplants are done for conditions like viral hepatitis, liver cancer, cirrhosis from alcohol or other toxins, and congenital abnormalities.
And pancreas for diabetes.
Right. For type 1 diabetes, whole pancreas transplants are sometimes done, often along with a kidney transplant. But a newer approach involves isolating the insulin-producing islet cells from a donor pancreas and transferring just those cells, often into the recipient's liver via the portal vein. These islet cells can establish themselves there and start producing insulin. Over 50% of recipients of islet transplants have become insulin independent for up to 2 years, which is a major improvement in quality of life, although long-term function and the need for immunosuppression remain challenges.
And skin grafts.
Most skin transplants are autografts, using the patient's own skin, for example, to cover burns. Foreign skin, allografts, is sometimes used as a temporary biological dressing for severe burns, but it's eventually rejected. There are some interesting innovations, though, like Brazilian scientists pioneering the use of sterilized tilapia fish skin as a dressing for burns. It seems to adhere well, protect the underlying tissue, reduce pain, and maybe speed up healing.
Are there other frontiers? Like brain grafts?
There's research into intracerebral neural cell grafts, for instance, for Parkinson's disease, trying to replace the dopamine-producing cells. This raises questions about using embryonic versus adult stem cells. And across all organ types, a major factor influencing success is the tissue damage that inevitably occurs during organ retrieval, preservation, and transfer ischemia, release of damage signals. Actually, this damage itself fuels inflammation and can worsen rejection. So improving organ preservation techniques is key.
Do they ever transplant multiple organs together?
Yes. Sometimes combined transplants are done, like kidney and pancreas for diabetic kidney failure, or heart and lung. Interestingly, sometimes these synergistic transplants seem to have better odds of success than transplanting one organ alone. One hypothesis is that transplanting more donor tissue, including the blood cells resident within the organs, might actually help boost tolerance mechanisms, perhaps by providing more donor APCs or regulatory cells.
Okay, so the demand is high, the challenges are immense. Once a foreign organ, an allograft, is actually in place, what happens immunologically when the recipient's immune system recognizes it as them? What are the stages of rejection?
Well, as we've hinted, allograft rejection isn't just a random attack. It always displays the key attributes of adaptive immunity: specificity and memory. The rate of rejection can vary. Skin grafts, for example, being highly immunogenic, are generally rejected faster than kidney or heart allografts.
How do we demonstrate specificity and memory in rejection?
The classic experiments involve sequential skin grafting in mice. If a mouse of, say, strain A receives a skin graft from a genetically different strain B mouse for the first time, it undergoes first set rejection. The graft initially looks okay, gets blood vessels growing into it, revascularizes over 3-7 days, but then inflammatory cells infiltrate, the blood supply decreases, the tissue starts to die, necrosis, and rejection is usually complete by about 12-14 days. This is a primary immune response.
Okay, and the memory part.
If you then take that same strain A mouse after it has rejected the first graft and give it a second skin graft from another strain B mouse, it triggers a second set rejection. This is a secondary memory response. The rejection happens much faster, usually complete within just 5-6 days. This acceleration is due to the recall activation of graft-specific memory T and B cells generated during the first rejection.
And how do we know it's specific to strain B?
You can prove this specificity easily. If at the same time you place the second strain B graft You also place a graft from a third, unrelated strain, say strain C, onto that same strain A mouse. The strain C graft will be rejected according to the slower first set kinetics, 12-14 days, while the second strain B graft undergoes that accelerated second set rejection, 5-6 days. It clearly shows the memory is specific to the antigens of strain B.
Okay, so graft rejection follows this pattern. In the clinic, how does rejection manifest? What are the different types based on timing and mechanism?
The clinical course of rejection is also fairly predictable, and we categorize it mainly by timing and the underlying immune mechanisms. The main types are hyperacute, acute, and chronic rejection.
Let's start with hyperacute rejection. You said that's the fastest.
Yes, hyperacute rejection is the most immediate and dramatic form, occurring literally within minutes to the first 24 hours after the transplant vessel anastomosis, when blood flow is restored to the graft.
Minutes. What causes such a rapid attack?
It's caused by those pre-existing host serum antibodies we discussed earlier, the ones detected by the cross-match test. These antibodies are usually specific for donor ABO blood group antigens or for donor MHC class alloantigens expressed on the graft endothelium. It's also often called antibody-mediated rejection, AMR.
And what happens when those antibodies bind?
When these pre-existing recipient antibodies bind to the foreign antigens on the endothelial cells lining the graft capillaries, it immediately triggers the complement cascade and attracts neutrophils. This leads to severe inflammation, endothelial cell damage, platelet aggregation, thrombosis, blood clots forming in the capillaries, and complete blockage of the blood vessels. The graft essentially never becomes properly vascularized and dies very quickly from lack of blood flow and oxygen.
Where do these pre-existing antibodies come from again?
Antibodies against ABO antigens are naturally occurring, likely from exposure to similar sugars on gut bacteria. Antibodies specific for MHC alloantigens, however, are acquired through prior sensitizing events. Past blood transfusions, previous pregnancies, certain infections that elicit cross-reactive antibodies, or previously rejected transplant. Thankfully, because we now do careful pre-transplant testing, ABO matching and cross-matching true hyperacute rejection is actually quite rare today.
Okay, so hyperacute is largely preventable. What about acute rejection? That happens later, right?
Yes, acute rejection typically occurs weeks to months after the transplant in recipients who didn't have pre-existing immunity to the graft. It involves the recipient's T cells recognizing and responding to the foreign antigens in the graft. It basically unfolds in two phases, much like a primary adaptive immune response or a hypersensitivity reaction, a sensitization phase and an effector phase.
Okay, what happens in the sensitization phase?
During sensitization, the recipient's CD4 plus and CD8 plus T cells encounter and recognize the foreign antigens, both major MHC and minor histocompatibility antigens expressed by the cells of the graft, and then they proliferate and differentiate into effector cells.
How do the T cells see these foreign antigens?
This recognition can happen through two main pathways. First is direct presentation or direct allo recognition. In this case, antigen-presenting cells, APCs, especially dendritic cells that are carried within the transplanted organ tissue, so-called passenger leukocytes, migrate out of the graft to the recipient's draining lymph nodes. There, they directly present their foreign MHC molecules, both class I and class II, complex with peptides, to the recipient's T cells.
Okay, donor APCs talking directly to host T cells. What's the other way?
The second pathway is indirect presentation or indirect allo recognition. Here, the recipient's own APCs take up cellular debris or proteins shed from the donor graft cells. They process these foreign proteins, including the foreign MHC molecules, and then present peptides derived from them on their own self-MHC molecules, class II for CD4 plus T cells, and class I via cross-presentation for CD8 plus T cells.
So the host APC presents bits of the foreign graft.
Exactly. Both direct and indirect pathways likely contribute to T cell sensitization. And this process is often amplified because the tissue damage and inflammation inevitably associated with the surgery and organ transfer itself release danger signals, NEPs, that act as natural adjuvants, promoting APC activation and T cell responses.
Interestingly, I've heard that sometimes giving a transfusion of donor blood before the transplant can actually help acceptance. That seems counterintuitive if it sensitizes.
That's a fascinating paradox, known as the transfusion effect. Yes, while transfusions can sensitize and cause antibody formation, under certain conditions, particularly if given deliberately without strong inflammatory signals, prior exposure to donor antigens can actually induce tolerance or hyporesponsiveness, perhaps by favoring the generation of regulatory cells or energy. The exact mechanisms are still debated, but it highlights how context matters.
Okay, so after T cells get sensitized, what happens in the effector phase of acute rejection?
The effector phase typically manifests about 7 to 10 days later or longer depending on the level of immunosuppression. It's characterized by a significant influx of leukocytes into the graft tissue, especially T cells, both CD4 plus and CD8 plus, and macrophages.
And T cells are definitely the key players here.
Absolutely crucial. We know this from classic experiments. For example, nude mice, which lack a thymus and therefore functional T cells, cannot reject allografts or even xenografts. And if you take T cells from a mouse that has already rejected a graft, an allograft prime mouse, and transfer them into a naive mouse of the same strain, that recipient mouse will now mount an accelerated second set rejection of a graft from the same donor strain. It proves T cells mediate rejection.
What does the rejected tissue look like microscopically?
Histologically, the infiltrate often resembles a delayed type hypersensitivity, TTH response. You see lots of infiltrating lymphocytes and macrophages, edema, and tissue damage, driven largely by cytokines released by activated T helper cells, which recruit and activate macrophages. Direct killing of graft cells by cytotoxic T lymphocytes, CTLs, recognizing foreign MHC class I via either direct or indirect pathways, also contributes significantly to the damage.
Do CD4 plus and CD8 plus T cells work together?
Yes, studies have clearly shown they collaborate. A key study by Cobbold and colleagues demonstrated that while depleting either CD4 plus or CD8 plus T cells alone only modestly prolonged graft survival, depleting both populations together resulted in dramatically expanded, sometimes indefinite, graft survival in mice.
And what specific cytokines are driving this acute T cell mediated rejection?
Cytokines secreted by T helper cells are central. Th1 cytokines, particularly IL-2, which drives T cell proliferation, and IFN gamma, which activates macrophages, increases MHC expression on graft cells and promotes CTL development and IgG antibody switching, are considered crucial mediators. Many other cytokines that encourage MHC expression also increase during rejection episodes, making the graft even more visible to the immune system. Th2 cytokines like IL-4, IL-5, IL-6, and increasingly recognized Th17 cytokines like IL-17 are also implicated. As we mentioned, recent studies showing that neutralizing IL-17 can prolong cardiac allograft survival in mice have drawn a lot of interest to the role of Th17 cells in rejection too. And let's not forget, antibody-mediated rejection, AMR, driven by T-cell-dependent alloreactive B cells making anti-donor antibodies, can also contribute significantly to acute rejection episodes, not just hyperacute.
Okay, finally, what about chronic rejection? You mentioned this is the one that's still really tough to beat.
Yes, chronic rejection is indeed the most significant long-term challenge in transplantation today. It can develop months, or more typically years after the transplant, often after any acute rejection episodes have been successfully controlled. It involves both humoral antibody and cell-mediated mechanisms, but the pathology looks different from acute rejection.
How does it look different?
Instead of a rapid inflammatory assault, chronic rejection is characterized by a slower, more insidious process. Key features include fibrosis, the thickening and scarring of the graft tissue, and narrowing of the graft arteries, graft arteriosclerosis, due to proliferation of smooth muscle cells in the vessel walls. This gradually reduces blood flow within the organ, leading to ischemia and a slow, progressive decline in its function, eventually leading to graft failure.
And immunosuppression doesn't work well against it.
Unfortunately, not very well. Despite the impressive progress made against hyperacute and acute rejection using current immunosuppressive drugs and better matching, much less progress has been made against chronic rejection. You can see this in the long-term survival statistics. For kidney transplants, for example, one-year patient survival rates might approach 97%, which is amazing. But even with kidneys from living donors, the 10-year graft survival rates can drop to as low as 60%, largely due to chronic rejection. Current immunosuppressive drugs often do little to halt or reverse chronic rejection once it starts, and it frequently necessitates going back on the waiting list for another transplant.
What drives chronic rejection? Is it antibodies again?
Antibodies seem to play a major role in many cases. It's estimated that in up to 60% of cases of chronic allograft dysfunction, the patients have detectable anti-donor antibodies, particularly against HLA molecules. This suggests that antibody-mediated mechanisms, perhaps involving complement or ADCC, are significant contributors. However, T-cell mediated responses and non-immune factors related to the initial injury or chronic inflammation likely contribute as well. Unfortunately, the specific triggers or underlying mechanisms that initiate and sustain chronic rejection are still not well understood, making it a critical focus for future research.
Okay. So, given these rejection processes, how do doctors actually manage the immune response? How do they keep the immune system from attacking the new organ? And what are the trade-offs involved with the drugs they use?
Right. As we established, transplanting an allograft between genetically different individuals always requires some degree of immune suppression in the recipient for the graft to survive long term. The major downside, however, is that most of the immunosuppressive treatments we currently use are relatively nonspecific.
Meaning they suppress the whole immune system.
Exactly. They suppress all immune responses, not just the ones specifically targeting the allograft. And this generalized immunosuppression leaves the recipient highly vulnerable to infections, which, importantly, remains the most common cause of death for transplant recipients. It also increases the long-term risk of developing certain cancers, because the immune system normally helps keep cancerous cells in check.
Are there other side effects?
Yes, many of these drugs also have significant off-target toxicities because they affect other rapidly dividing cells in the body, like the cells lining the gut or the stem cells in the bone marrow. This can lead to side effects like nausea, diarrhea, anemia, or low white blood cell counts. Some drugs also have specific organ toxicities, like kidney damage, or can cause metabolic problems like hypertension, high cholesterol, or metabolic bone disease. So, managing immunosuppression is a constant balancing act for transplant physicians and patients trying to prevent rejection without causing unacceptable toxicity or oversuppression.
What are the main strategies or types of immunosuppressive therapies used?
Broadly, we can think about a few categories. One older strategy, though not commonly used for most solid organ transplants today, is total lymphoid irradiation, TLI.
Irradiation, like radiation therapy.
Yes, this involves using X-rays to specifically target and eliminate lymphocytes in the recipient's major lymphoid organs, the thymus, spleen, lymph nodes, just before the transplant surgery. It's often used prior to bone marrow or hematopoietic stem cell transplantation.
How does it help?
By eliminating the existing mature lymphocytes. The bone marrow itself is usually shielded, so new lymphocytes can still be generated after TLI. The idea is that these newly formed lymphocytes, developing in the presence of the graft antigens, might be more likely to become tolerant to the graft. TLI can also sometimes be used to treat established GVHD after bone marrow transplant.
Okay. What about the drugs? You mentioned the first generation generalized immunosuppressants were game changers.
Absolutely. The introduction of drugs like azathioprine in combination with corticosteroids in the 1960s, based on work by Robert Schwartz, William Damashek, and Joseph Murray, truly revolutionized transplantation. It dramatically increased allograft survival rates. Murray, along with Gertrude Elian and George Hitchings, who developed azathioprine, shared a Nobel Prize for this work.
How does azathioprine work?
Azathioprine, often sold as Imuran, is a potent mitotic inhibitor. It gets converted in the body to 6-mercaptopurine, which interferes with the synthesis of purines, essential building blocks for DNA. By blocking DNA synthesis, it preferentially kills rapidly dividing cells, including proliferating B and T lymphocytes that are responding to the graft antigens.
Are there other drugs like that?
Yes, other mitotic inhibitors like cyclophosphamide, which is an alkylating agent that damages DNA, and methotrexate, which is a folic acid antagonist that also blocks purine biosynthesis, are sometimes used in certain transplant or autoimmune settings. However, because they hit all rapidly dividing cells, they have significant side effects on the gut lining, liver, and bone marrow.
And corticosteroids, they're used a lot too, right?
Yes, corticosteroids like prednisone or dexamethasone are potent anti-inflammatory drugs and are a cornerstone of most immunosuppressive regimens, especially for treating acute rejection episodes. They work through multiple mechanisms, but a key one is repressing the activity of NF-alpha, a crucial transcription factor that controls the expression of many genes involved in immune activation and inflammation, including genes for cytokines like IL-1, IL-6, and TNF-alpha.
But I understand that drugs derived from fungi really changed the game again, offering more specificity.
Yes, the discovery and development of certain fungal metabolites, starting with cyclosporin A, CSA, in the late 1970s, represented another major leap forward. Later came FK506, tacrolimus, and rapamycin, sirolimus. These drugs, although chemically distinct, share a remarkable ability to more specifically block the activation and proliferation of resting T cells, rather than just killing all dividing cells.
How do they block T cell activation?
CSA and tacrolimus work by inhibiting calcineurin, a phosphatase required for activating NFA, another key transcription factor needed to produce IL-2. Rapamycin works differently, inhibiting a pathway involving mTOR, which is crucial for cell cycle progression in response to IL-2 signaling. By preventing the transcription of IL-2 and its high-affinity receptor, IL-2I or CD25, or by blocking the response to IL-2, these drugs effectively shut down T-cell proliferation, which is central to initiating graft rejection.
And these are widely used now.
Absolutely. They became the mainstay of immunosuppression for most solid organ and bone marrow transplants, leading to further significant improvements in graft survival. CSA was revolutionary, though it can cause kidney toxicity. Everolimus and sirolimus are even more potent, often 10 to 100 times stronger than CSA on a weight basis, allowing for lower doses and sometimes fewer side effects, although they have their own toxicity profiles too. Combination therapy using low doses of several drugs is now standard practice.
The ideal, of course, would be completely antigen-specific immunosuppression, only targeting the anti-graft response. We're not quite there yet, but what are the more targeted approaches being developed or used?
That's absolutely the goal, to induce donor-specific tolerance while leaving the rest of the recipient's immune system fully intact to fight infections and cancer.
Yeah.
While we haven't fully achieved that holy grail yet, several more targeted strategies have emerged, primarily using monoclonal antibodies, ABs.
Antibodies as drugs.
Yes. Using genetic engineering, scientists can now create humanized or fully human mAbs, which overcome problems like immune responses against the older mouse-derived antibodies. These mAbs can be designed to target specific molecules or cell types involved in rejection.
How do they work? Depleting cells.
Some work by depleting specific immune cell populations. For example, antithymocyte globulin, ATG, is a polyclonal antibody preparation made by immunizing animals with human T cells that broadly depletes T lymphocytes. OKT3, an older anti-CD3 mAb, also rapidly depletes mature T cells, though it causes significant side effects. Newer approaches might use antibody toxin conjugates to deliver a killing blow more specifically. And rituximab, an anti-CD20 mAb, is used to deplete B cells, particularly to treat or prevent antibody-mediated rejection, AMR.
Can they block function instead of depleting?
Yes, other mAbs work by blocking key steps in immune activation. For example, basilicimab is an AB that targets CD25, the alpha chain of the high-affinity IL-2 receptor, which is upregulated on activated T cells. By blocking IL-2 binding, it inhibits the proliferation of these activated T cells. A potential drawback, however, is that regulatory T cells also express high levels of CD25, so it might inadvertently affect these beneficial cells too. mAbs, again, and T-cell specific markers can also be used to pre-treat donor bone marrow to help prevent GvHD.
Since cytokines like TNF-alpha are involved in rejection, are there therapies targeting them?
Yes, targeting cytokines is another strategy. mAbs against pro-inflammatory cytokines like TNF-alpha have been explored. Anti-TNF therapies, widely used for autoimmune diseases like RA, have shown some promise in animal models for prolonging bone marrow transplants and reducing GvHD severity. mAbs against IFN-gamma or the IL-2 receptor itself have also been investigated.
What about interfering with that co-stimulation step, signal 2 for T cells? That sounds like a precise way to disarm them.
It is a very attractive strategy. Remember, T-cell activation requires both the TCR signal, RAISINS-1, and co-stimulation signal 2, typically via CD28 binding CD80-DO86. If a T-cell receives signal 1 without adequate signal 2, it often becomes anergic, unresponsive, or might even differentiate into a regulatory cell.
So can we block signal 2?
Yes, we know that CTLA-4 is a natural inhibitor that binds CD80-TS6 with much higher affinity than CD28. So scientists cleverly created a soluble fusion protein called CTLA-4Ig. It combines the extracellular domain of CTLA-4 with the constant region Fc of a human IgG1 antibody.
And this acts like a decoy.
Exactly. This therapeutic drug, which comes in two main forms, abatacept torrentsia, and a second-generation version with even higher affinity called belatacept nulogics, binds tightly to CD80 and CD86 on antigen-presenting cells. By doing so, it physically blocks them from engaging CD28 on T cells, thus preventing the delivery of signal 2. This leads to T-cell energy or hyperresponsiveness, specifically towards the antigens being presented, including the graft antigens. Belatacept in particular is FDA approved specifically for preventing kidney transplant rejection and represents a significant step towards more targeted immunosuppression.
Okay, so with all these improving drugs, is it actually possible to get the body to just fully accept a foreign organ without needing any of these powerful, lifelong immunosuppressive drugs? Is there a holy grail of transplantation tolerance?
Yes, achieving transplantation tolerance defined as the long-term survival of an allograft in the absence of ongoing immunosuppression, that is absolutely the ultimate goal, the holy grail of the field.
Does it ever happen naturally, or can we induce it?
It can happen, but it's rare spontaneously in humans after standard transplantation. However, there are situations that naturally favor tolerance. For instance, tissues that intrinsically lack significant allonogens, like cartilage or heart valves, don't typically elicit strong rejection.
What about those immunologically privileged sites?
Right. Certain anatomical locations in the body are considered immunologically privileged sites. These include the anterior chamber of the eye, the cornea, the uterus, crucial for pregnancy tolerance, the testes, and the brain. These sites seem to have limited access for immune cells, perhaps due to unique vasculature, lack of conventional lymphatic drainage, or the presence of active immunosuppressive factors locally. This relative isolation reduces the chances of T-cell sensitization against antigens expressed there. That's why corneal transplants, for example, are highly successful even without rigorous matching or strong immunosuppression. And researchers are trying to exploit this, for instance, by encapsulating pancreatic islet cells within semi-permeable membranes before transplanting them. The membrane physically blocks immune cells, but allows insulin out.
Beyond these special sites or tissues, can we actually induce a state of tolerance clinically for a regular organ transplant?
That's the major focus of current research, trying to actively induce donor-specific tolerance. Nature provides some clues. We mentioned the non-identical twin cattle sharing a placenta observed by Ray Owen back in the 1940s. They were naturally tolerant to each other's tissues. This led Billingham, Brent, and Medawar to their Nobel Prize-winning work in mice, showing that exposure to allonogens very early in life, during immune system development, could induce lifelong specific tolerance.
And the microchimerism from pregnancy.
Yes, the persistence of small numbers of fetal cells in the mother, or maternal cells in the child, for decades might also contribute to maintaining a state of mutual tolerance, though the exact role is still being worked out.
So how are researchers trying to induce tolerance clinically now?
One major approach focuses on inducing a state of mixed hematopoietic chimerism. This means creating a situation where both donor and recipient hematopoietic stem cells coexist stably in the recipient's bone marrow and give rise to immune cells.
How is that achieved?
Various protocols are being tested. Some involve quite intensive treatment for the recipient first, like total myeloablative therapy, using radiation or chemotherapy to wipe out the recipient's bone marrow, followed by a transplant of donor bone marrow before or at the same time as the solid organ transplant from the same donor. The idea is that the recipient's developing immune system will mature in the presence of donor antigens derived from the donor HSCs, leading to central tolerance. Or modified, less harsh, non-myeloablative conditioning protocols are also being developed and show promise in achieving stable mixed chimerism and improved graft outcomes with reduced toxicity.
What are the mechanisms behind tolerance in these chimeric individuals?
It's thought to involve multiple mechanisms. Certainly, central deletion of newly developing alloreactive T cells in the thymus, recognizing donor antigens presented by donor-derived APCs, likely plays a key role. But there's also strong evidence for the enhancement or induction of regulatory T cells, TREGs, specific for the donor antigens.
TREGs again. They keep showing up.
They are absolutely central to tolerance, yes. Studies of the rare human transplant recipients who have become operationally tolerant, meaning they've successfully stopped all immunosuppression years after transplant without rejecting their graft, often find increased numbers or activity of TREGs, particularly those specific for the donor's HLA type.
So TREGs are key to maintaining tolerance long term.
It strongly appears so. These TREGs likely inhibit any remaining alloreactive effector cells through direct contact, cytokine secretion, like TGF-beta, IL-10, IL-35, and other mechanisms we discussed. This has led to really exciting, cutting-edge therapeutic approaches.
Like what?
Like actually isolating a patient's own TREGs from their blood, expanding these TREGs massively ex vivo in the lab to generate billions of them, and then infusing this large population of TREGs back into the patient around the time of their transplant surgery. The hope is that this boost of regulatory cells will help establish and maintain tolerance to the graft, potentially allowing for reduced immunosuppression later on. Clinical trials testing this TREG adoptive therapy are underway. If we connect this to the bigger picture, leveraging the body's own sophisticated regulatory mechanisms like this is where the real precision and promise of future therapies lie.
Wow. So let's try to wrap this up. What does this all mean? We've really journeyed from the body's innate, incredible ability to recognize self and tolerate the good guys like our microbes to the complex and sometimes devastating breakdowns in that system that lead to autoimmunity. And then finally, exploring the incredible medical leaps we've made in transplantation, where that very self-recognition system becomes the biggest hurdle. It really is a story of exquisite balance, isn't it? The profound consequences when that balance is disrupted, and our relentless scientific and medical efforts to understand and restore it.
It truly is. And our understanding, as you said, has genuinely blossomed. From initially viewing tolerance as just simple ignorance or deletion, we now appreciate its incredibly nuanced active mechanisms, things involving evasion, yes, but also elimination, and critically, this active engagement by various regulatory cells, especially TREGs. And it's precisely this depth of understanding that's allowing for the development of the novel, more targeted treatments we discussed today.
Right, from those co-stimulation blockers like belatacept to trying to harness TREGs directly.
Exactly. It also highlights how deeply interwoven these three areas, tolerance, autoimmunity, and transplantation, really are. The immune system's fundamental drive to maintain self-tolerance is simultaneously our most vital protector and the greatest barrier we face when trying to achieve transplant success.
So as we wrap up this deep dive, here's something for you, our listeners, to consider. Our immune systems are just this incredible testament to evolutionary fine-tuning, right? A constant dynamic negotiation between vigilance against threats and tolerance towards self and harmless antigens. Given this incredible daily battle for self-tolerance that's happening inside all of us, what might be the next surprising environmental factor we discover? Something, like gut microbes or maybe even salt intake that tips the scale towards autoimmune disease.
Or conversely, perhaps unlocks a new path to achieving that holy grail perfect drug-free tolerance for transplanted organs. The field's moving so fast, it's exciting to think about what discoveries lie just around the corner.
Absolutely. There's still so much to explore in immunology.